welcome Dr. Miguel Fortuna. He's a computational evolutionary ecologist here at the EBD. He obtained his doctorate degree here at the Doñana Biological Station in 2008. And then he moved to the University of Princeton, where he stayed for 3 years in the United States. Then he came back to the EBD as a postdoc again. And then he also did a 4-year postdoctoral stay in the University of Zurich. And now he's back at the EBD as a Ramón y Cajal researcher. So yeah, whenever you're ready.
Okay. Thank you very much, Tere, for the introduction. So instead of talking about my previous work, I'm going to take advantage of this opportunity to give you a quick overview of the long-term research line that I would like to carry out here in the next years here in the EBD if I finally get a permanent position. So I'm not going to present any results, just the main idea behind the project, so that hopefully in a few months you still can identify what I'm working on. As a postdoctor, as a Ramón y Cajal researcher, I started to set up my own lab at the beginning of last year. Because in January 2020, we were warned about the worst pandemic in the last century. You know about that. But COVID-19. But for many years, we've been also warned about another pandemic, a slow motion pandemic that is even worse than the COVID-19, which is the antibiotic resistance crisis. Bacteria are developing resistance to many, if not all, the antibiotics that we have available in the market. So it is estimated that 700,000 people die annually in the world because of resistance of bacteria to the antibiotics. And if we don't do anything, by 2050, 10 million people could die annually in the world, which is twice the number of people who have unfortunately passed away. So it's a very serious problem, the one that we have. You all know that, and I'm just showing the way, or one of the ways that bacteria develop or evolve resistance to the antibiotics. A bacterial population has many variants. The genetic makeup of a bacterial population is quite diverse, it can be quite diverse. So when we got a bacterial infection and we take an antibiotic, some of those bacteria might be already resistant to that antibiotic. So again, the bacteria that was resistant to the antibiotic will take over the entire population. I'm just showing this to you to remind that antibiotic resistance is an evolutionary process. And as such, we cannot stop, right? Because we cannot prevent evolution to happen. But, and that's the main idea behind the research line or the projects I would like to conduct. I think that we can at least harness evolution. I think we can control, we can guide the evolutionary trajectories of populations towards a decided state. That's the main idea. It's like riding a wild horse. You might not be able to stop it, but at least you can guide it towards a safer pathway. And that's something that I think we could do, or we could apply that idea in this field of to solve this problem of antibiotic resistance. So what can we do when we take an antibiotic and
the bacterial infection is resistant to that antibiotic. We can try and take a different one, right? But what can we do when all the antibiotics that are available in the market don't kill the bacteria? So we cannot do anything. We cannot do anything. People are dying because of that. So we need alternatives to the use of antibiotics. And one promising alternative is called phage therapy, which consists of using the natural enemies of bacteria, viruses known as bacteriophages or simply phages, to combat or as therapeutic agents to fight against bacterial infections that have been resistant to the antibiotics. In contrast to antibiotics, which can kill many different bacteria, phages are very specific. They can only infect a single species of bacterium or even a single strain within a species of bacterium. And that's good for us because they are not going to kill the good bacteria that we have, for example, in our guts or in our microbiomes, right? But as bacteria do with antibiotics, bacteria can evolve to escape from being infected by the phages. So, and this is a cartoon that shows you the same process that we saw previously. So in a population of bacteria, some mutants might have changed the molecules that they show on the surface and that are used by the phages as receptor to bind it and to inject the nucleic acid, right? So at the end, we will have, again, as we saw with antibiotics, a population that is resistant to both the antibiotic and also to the phage that we have used. So what can we do then? We have to find and identify a new phage that will be effective against that new strain of the bacteria, right? Which complicated this, the application, the real application of phage therapy. Is there any solution to solve this problem? Well, I think, and this is an important difference between an antibiotic and a phage, the advantage is that a phage can co-evolve with the bacteria. The phage can counterbalance the defense traits that the bacteria have evolved to escape from being infected by the phage. So the idea then, and I think we as ecologists and evolutionary biologists can play a major role in contributing with our expertise to the advancement of this new field of phage therapy. Because the idea is to find a way or to do something that so that the phages could cope much better with the evolution of bacteria. So the idea is to find mechanisms that promote phage coevolution, so that we inject a phage to treat a bacterial infection, and we could be confident that if the bacteria tries to escape from being attacked by the virus, by the phage can counterbalance the evolution of the bacteria. So it can still infect those new variants of the bacteria that might emerge as a consequence of the selective pressure imposed by the phage. And that's the idea behind this project. The idea is to find mechanisms trying to play with ecology, trying to engineer ecological interactions, playing with the matrix of competition for resources among the bacteria, to make or to promote phage coevolution, to make the phages more evolvable. This has to do with the evolution of the evolvability. How can we increase the evolvability of the phages? So, and that idea came to my mind when I was in Switzerland. We were working, or we were exploring how coevolutionary dynamics shapes the structure of bacteriophage infection networks in the lab in collaboration with some microbiologists. And while working on this paper, or when I understood what the microbiologists were doing, doesn't work. Yeah. So the microbiologists set up or set a coevolutionary experiment in the lab. So they use Pseudomonas fluorescens as a bacterium and one of its phages. And they, wow, too fast. Well, doesn't matter. So they put in a flash disk the bacteria and the phage and let them coevolve for 24 days. Right? They did 2 treatments. In one of them, they put a lot of nutrients for bacteria to grow, and in the other, it was a poor nutrient medium. And they did 6 replicates for each treatment. So basically, every four days, they took a sample of the co-evolving population and isolated 20 phages and 20 bacteria. This is what we see here. Phages are represented here as rows and bacteria as columns. So they, at time t equal 1, at day 4, they isolated 20 phages and 20 bacteria, right? And they did the same for each time step. 24 days, 6 time steps. So we built this infectivity matrix. And we have one matrix for each replicate, of course. So what they did after, once the experiment finished, they performed pairwise tests between each phage and each bacterium that they previously isolated. So for this time step, they performed 20 times 20, 400 pairwise crosses to see whether the phage was able to infect that bacteria, right? And they did that for each time step. And they also did, and that's, I think that's fantastic, they call that time shifts. So they also check whether phages isolated in the future could be able to infect bacteria from the past. For example, if we look here, they check whether these 20 phages that were isolated at time t equal 2 were able to infect these 20 bacteria that were isolated at the previous time step. Do you follow with me? For example, here, 20 phages isolated at time t equal 3 against bacteria isolated at time t equal 1 and at time t equal 2, right? And they did the same for this region of the infection matrix. So in total, they performed 20 times 6, 120 times 120. More than 40,000 pairwise tests. And when I was thinking on this, I realized, well, if we pay attention to this sentence here, phages from the past and bacteria from the future. So if we find a lot of interactions here of ones, which means that phages from the past can infect bacteria that were isolated at later points in time, those phages would be pre-adapted to the bacteria because they will be able to infect bacteria they never have been exposed to. And I link that to that idea of what can we do to fill this part of the matrix with ones. What can we do to make a phage be able not just to infect the bacteria they are co-evolving with at the same time, but to be able to infect bacteria or the strains of the bacteria that will evolve in the near future? Yep. So let me show you the result of that particular study. So now we are going to plot the infection probability, which is the fraction of ones over the total number of pairwise interactions for each of the three regions of the matrix that I showed you previously, right? And the resistance probability, which is the complementary measure, the fraction of zeros, right? So at low resources, in the low resources treatment, if we take this as these two values as the reference values, because they are bacteria and phages that coevolve at the same time step, right? This is the probability of a phage to infect a bacteria from which it's coevolving at the same time. If we look at here, for example, which is the bacteria from the future and the phages from the past, whether phages could infect the bacteria that will be in the population in the future. We see that the resistance probability is lower than for contemporary bacteria, which means that bacteria that evolves have lost the resistance traits that they have evolved previously. That's why the resistance of the bacteria to phages from the past is lower than from contemporary bacteria. This is a classic example of fluctuating selection dynamics. So you develop resistance traits and infectivity traits, but you lose them when you don't need it, and you evolve another ones, right? And this is a scenario this scenario is quite different from what we see at high resources. So when bacteria have a lot of resources, we observe an arms race coevolutionary dynamics. And if we look again, we take as reference the resistance probability of the bacteria coevolving with phages at the same time step. Now, bacteria from the future are still or even much resistant to phages from the past. This means that bacteria kept the defense, the resistance traits that it evolved, right? And the same happens if we look at the infectivity of the phages. Phages from the future and bacteria from the past. We see that phages that have evolved, let's say at time t equals 6, are still able to infect the bacteria that were at the population at time t equal 1, because they still have the infectivity traits that they evolve. So it's an arms race classical scenario in which you have bacteria accumulate resistance traits and phages accumulate infectivity traits. So let me go back, because the idea, I'm having problems with this, it's too slow. So it doesn't matter if you remember the point on the left in the arms race coevolutionary dynamics, which represented the infectivity of phages from the past and bacteria from the future, was very low. So phages were not pre-adapted to the bacteria that they will encounter in the future. And that exactly what we want to do is to increase the infectivity of those phages. So instead of having that low value of infectivity, we would expect, or we should find a way to increase that infectivity so that the phages that are co-evolving with the bacteria could infect them even if they evolve new resistance traits. So that's the idea behind everything. The idea is that we can play, because that study shows that by modifying the ecology of the system, by manipulating the amount of resources available for bacteria, we can change coevolutionary dynamics. It's a proof of how we can work modifying the ecology of the system and drive the evolutionary outcome towards some points where we are interested in, right? So this is what we want to do. We would like to promote the evolvability of the phage, the ability of the phage to counterbalance the resistance traits that bacteria will evolve in order to escape from being infected from it. And we are planning to do this in 3 different stages. So in the first stage, we are planning to do this in computers, working with simulations and mathematical models. The second stage would be to apply what we are going to learn here in silico. The idea is to apply it in vitro in the microbiology lab. So all the ideas that we can get from this analysis, we can then move and try to evolve phages in the lab with real bacteria and phages. And a third stage that would be to test the efficacy of the phages that we will evolve in the lab in vivo using an animal model. So far, we are working on this stage. Now I'm going to focus on this part. So you will know that evolution can be studied in the wild, as Peter and Rosemary Grant, for example, do in the Galapagos Island with the Darwin finches. Evolution can also be studied in the laboratory. A paradigmatic example is the work by Richard Lenski. I don't know if you know his long-term evolution experiment using E. coli. It's very slow. Yeah, here you have Richard Lenski. So the bacteria are evolving over more than, right now, over more than 60,000 generations, more than 25 years. He started his experiment 25 years ago. But perhaps a few of you know that evolution can also be studied in computers. And I'm not meaning that, I don't mean that you can use your computer to study evolution. Of course you can do. I mean that evolution can take place within the computer as a different, a complementary evolutionary system. My computer is too old and yeah. So we are using in our lab a digital evolution approach. We are using an artificial life software platform that was developed in the California Institute of Technology, but now the developers are working at the Beacon. It's an NSF center based on Michigan State University. That software platform is called EBD. And in EBD, this software platform, self-replicating computer programs mutate and evolve within a user-defined computational environment. A digital organism, which is how we call these computer programs, consists of a circular list of instructions, codes, that represents its genome. you can see here a representation of a genome of one of those digital organisms. In this case, only 12 instructions long. It's a very short genome, right? The genetic language of these digital organisms consists of 26 instructions. You have here the list, don't pay attention to this, it's not relevant, that are represented by letters. So we have 26 different letters. You might think of biological organisms, we have 4 nucleotides, 4 letters. Here we have 26 instructions. The important thing is that some of these instructions, some of the genetic language is involved in the replication process, which is the only means a digital organism can pass its genetic material to future generations. They replicate asexually, they emulate what bacteria do. And now in this short animation, you see how one of these genomes makes a copy of itself. During the copy process, some errors might happen, some mutations. One instruction can be replaced by another, randomly taken from the instruction set, from that 26 instruction codes. Insertions and deletions are also implemented here and are also possible. In this case, the offspring is an exact copy of its father. There was no mutation in this case, no? The mutation probability is the only thing that you as a researcher set in this kind of experiments. You put the ancestral organism, single organism, you set up a mutation probability and let it evolve. Besides those instructions that are involved in the copy process, there are other instructions that are involved in taking binary numbers from the environment, store them into input buffers, and manipulate those numbers. So some instructions can manipulate binary numbers and do operations with them. And I'm going to briefly explain what I mean by that. So here, the arrow means the instruction flow. In this time step, the digital organism has executed the V instruction. In that one, the Y instruction, right? Here, you can see, I hope you can see here, 32 boxes, 2 input buffers with 32 boxes each. They represent the binary numbers that they take from the environment. Binary number is a sequence of ones and zeros. So white square means a 0, black square means 1, right? We have here one binary number and here another one. Now with some of these instructions, some of those ones, when they executed some of those instructions, they modify those numbers and place the result on 3 registers. And the important thing is that there is one instruction in that genetic language, the Y instruction, the input-output instruction, that when it is executed, it outputs the content of the BX register that it has in the previous step. So you see here, the output is a binary number. Red means ones, red means 1, a white square means 0. And this number could be the result of applying a Boolean function. In this case, the not and Boolean function, which returns 0 if and only if both inputs are 1. Otherwise, it returns 1. And you can see here that this output, this binary number, is exactly the result of a plugin. Sorry. I have to go back so that you can understand what I mean. You can have a genome as long as you want. This is just very short for visualization purposes, right? Okay. So you see here 1, right? When both bits at the same position are 1, it returns 0. Those ones, those white boxes, you see? Otherwise, it returns, in those cases, it returns 1, the red squares. So these organisms, what I mean is that these organisms have the ability to compute Boolean operations, which are going to determine the phenotype of these digital organisms. So a genome, a genotype, can encode a particular phenotype depending on the Boolean function it can compute with the instruction it harbors in its genome. We are working with 9 Boolean functions because we are only allowing the digital organism to store 2 binary numbers. If we allow it to store 3 binary numbers, we could have up to 77 different Boolean functions. So since we are working with 9 Boolean functions, the size of the phenotype space is 2 raised to the power of 9, 512 different phenotypes. So any digital organism can encode one of those 512 different phenotypes. For example, this is one phenotype, a phenotype in which the digital organism performs only the not and Boolean operation, but we can have phenotypes in which the organism performs the equal function, the OR function, the NAND function, or even all the 9 functions. That happens because a digital organism might have more than one input-output instruction in its genome, so it outputs more than one binary numbers, or it might have only a single input-output instruction, but that instruction is within an execution loop, so it is executed several times. And we've built a huge database of digital organisms in which we have the genotypes, the sequence of letters, the sequence which is translated to the sequence of instructions for each organism, and also the phenotype that they encode. We have thousands of organisms. And the picture is much more complicated than what it looks like, because digital organisms can show phenotypic plasticity as well. 2 organisms with the same genome can encode different phenotypes in different environments, because the environment determines the random binary numbers that they take. So the binary numbers that they take can modify the instruction flow, the instructions that are executed within the genome of a digital organism. So there is a strong phenotypic plasticity in these organisms, or they can be. There are also genomes that encode always the same phenotype, but you can also find phenotypic plasticity, although I'm not gonna explain this today. So the point is that we characterize this mapping between genotype and phenotype for a huge number of genotypes, and we found that the mapping is not trivial. It's not a direct mapping. It's a non-linear mapping. There is a lot of epistasis here. We also shed light on questions related with the evolvability and the robustness of these digital organisms and how they are related, which is something I'm using when I'm thinking about how to increase the evolvability of phages, right? That's why I'm talking about this previous work. So here I'm showing you that the digital organisms live physically in the memory of the computer. They occupy the cells of the memory of the computer. So here you see a lot of digital organisms, each one occupying a cell memory. Organisms with the same color represent or encode the same phenotype. And as I told you before, at the beginning of an evolutionary experiment in silico, you just place one of these organisms here, all the rest of cells are empty. You set a mutation probability and let it evolve. And this is not new. The digital software platform was developed 20 years ago. And some researchers, the developers and others, even Richard Lenski, the one who is working in the long-term evolution experiment with bacteria, has made important contributions using digital organisms. So they publish, they explore fundamental questions in evolutionary biology. You cannot study a particularity of the evolutionary process, but you can study general trends in evolutionary biology, and they publish their results in top journals like Nature and Science, but only focusing on evolution. So my contribution to this field was adding ecological interaction to this artificial life software platform. So we have implemented antagonistic interactions between hosts and parasites, which is something new that haven't been explored before. Now, some organisms can steal CPU cycles, can steal time of the processor of other programs that are their hosts to speed up its replication process, slowing down the replication process of this organism that they are parasitizing, let's say, right? And in this short animation, you see that a host that is being infected by a parasite replicates at a slower rate than a host that has been infected. So we are imposing a selective pressure for hosts to escape from being attacked or infected by the parasites. This system emulates the phage bacteria infection that we saw previously, right? Or that we are interested in phage therapy. But not all parasites can infect all hosts. It depends on the infection mechanism. We have implemented 3 infection mechanisms that correspond to the models widely used in the literature. One that emulates the gene for gene model, the matching alleles model, and the inverse gene for gene model, which is the one we are using. The inverse gene for gene model means that a parasite can infect a host if it performs at least one of the Boolean operations that the host performs. In the biological world, it would be analog to, for example, for a bacteria to evolve a molecule on its surface, and the phage that have their tail fibers can bind that receptor on the bacterial cell. So if the phage can match at least one of the receptors of the bacterial cell, it can infect the bacteria. That would be analogous to the mechanism of infection that we have implemented here with these digital hosts and parasites. And as I told you, in the case of if you run an evolutionary process, you just start with one ancestor, you set the mutation probability and let it evolve. In this case, for doing a coevolutionary experiment, you start with one parasite, one host, you set a probability of mutation for the parasite, a probability of mutation for the host, and let them co-evolve. And this is how looks like one of these coevolutionary experiments. You observe rapidly, each node represents a phenotype, a different phenotype for the hosts and for the bacteria, for the hosts and for the parasites. Hosts will always try to escape by developing or by performing Boolean operations that are not performed by the parasites. The parasites evolve new Boolean operations so that they can still infect the bacterium, and you observe a diversification in the phenotypes of the parasite and also on the phenotype of the bacteria. The size of the node is proportional to the abundance, to the number of organisms, the number of parasites encoding that phenotype. And you see how everything changes continually. So instead of having a picture of a coevolutionary process, as we normally have, if we go and see what happened in nature. We have the full picture. Since these experiments take place within the computer, we have access to a huge amount of data. We have information about the genome of each individual, the number of individuals encoding each phenotype, who infects whom, the interaction strength, the mutation that took place for each individual, the evolutionary history of the entire system. And the good thing is that you can manipulate this very easily. And this happens very quickly. You don't have to wait a month to observe how a coevolutionary process unfolds. You see this depending on the number of generation, but for 100,000 generation, it can take 12 hours, perhaps. So you can manipulate a lot this system, this coevolutionary system in silico. And hopefully, that's what I hope, what we learn by doing this experiment, we can later on apply to the lab. I think we can learn a lot of things here. We can learn how to make the phage or the parasite co-evolve with the host, because in some cases, that's another good thing that it has, you can do a lot of replicates and you can quantify how repeatable the coevolutionary process is, for example. And in some replicates, the parasite, the host escapes from the parasite, and the parasite doesn't co-evolve with the host and go extinct. So you can manipulate the system or the environmental conditions, the abundance, for example, or the nutrients available for the hosts, so that you make the parasite more likely to co-evolve and to keep and to track changes in the host population. So as I told you, we are working on this part, but we would like also to translate what we learn here to the laboratory at some point. And to do it, I'm planning, or I would like to work with the Staphylococcus aureus as the bacterium, which we know that is resistant to a lot, and some strains to all antibiotics that are available in the market. And it has another advantage that phages from very different families are able to infect these bacteria. For example, from the Podobiridae family, Myoviridae family, Syphobiridae family, which they all have different morphologies, different genome size, which, by the way, I think it's also important in promoting the evolvability of the phage. The genome size might play an important role here. And I think this is this would be a very good system to explore ways and mechanisms that would lead to a phage to increase its coevolutionary potential, so that it can track bacterial resistance. And a novelty of this in vitro approach is the use of devices that are called turbidostats. Each of these tubes is called a turbidostat. My colleague in Michigan is working with this in the lab, and it's been doing a fantastic work, because these devices, they are continuous culture devices. You put here a liquid, a medium for the bacteria to grow. You put the bacteria, put the phage. You have a sensor that measure the optical density continually, and you have different pumps that are connected to a computer, so that you can add medium and take out at the same time. So with the computer, you can say, I want to keep the density or the population abundance of the bacteria to that particular level, and you can keep that population abundance constant over time, at low level, at high level, because of the computer will add or take out liquid, so that the population abundance of the bacteria is always the same. You can have it at low abundance and high abundance, or you can have oscillations in the population abundance, which is the key, I think, to increase phage evolvability. So this is a novelty, I think, because it allows you to control evolution, to control ecology. And by manipulating the ecology of the system, you see how that changes the evolutionary outcomes. You can control the ecology of the system with these devices. And finally, if everything works out well, or you can always test the efficacy of the phages that you have evolved in the lab in vivo. We are planning to work with the silkworm larvae. A lab in Japan is working with this silkworm larvae, and it's a good model for logistic reasons, much better than vertebrates like mice. And they are working on it, with it, and they prove that it's a good model to test the efficacy of phages destroying or fighting against bacterial infection. So the idea is that you infect the silkworm larvae with a bacterium, 24 hours later, you inject the phage, and now you monitor changes in survival of the larvae. So you can test to what extent the phages that you have evolved previously in the lab are better in controlling or fighting against the bacterial infection. And I don't want to take much of your time. So this is work in progress. I'm still thinking about many processes here. And if any of you are interested, please, you can visit the webpage of the lab, talk to me, and I would be happy to take questions and also receive some feedback from you. So thank you very much. So what do you think about this idea of promoting coevolution? Yeah, Xavi.
Muchísimas gracias, Miguel. Bueno, pues vamos a empezar con la sesión de preguntas.
Well, okay, thank you, Miguel.
I think the microphone's on.
Ah, bueno, pues.
Yo te oigo, sí, no problema.
Sorry. Well, Miguel, thank you very much, and particularly thank you for bringing fresh thinking to EBD, which is, I think, fundamental for our long-term survival. I don't want to become too dramatic, but I think that it's quite important, you know, to detect some kind of fresh ideas. It's not, I mean, the theoretical background is coevolution networks, which is something that in here with people are doing, right, and quite successfully. So a bunch of questions come up to my mind, but I have only one. So are you considering as well trade-offs? I mean, for example, if a mutation enhances infectability, perhaps it decreases lethality. And, you know, when a trait evolves, it means that it may have a cost.
Are you talking about the trade-offs, right?
Right.
Yeah.
And that's, it's not clear to me whether, you know, all these combination of mutations and the way in which you generate new phenotypes, there are some kind of forbidden relationships between mutations, because that can be one of the keys, you know, to add realism to the models.
You mean in the in silico approach?
Right.
Yeah.
Right.
In silico approach, it's very interesting because it happens what we observe in biological organisms. There is a trade-off between performing Boolean operations, that is developing or evolving new traits, if you want, and the replication rate. Because for computing Boolean operation, normally the digital organism must have to execute more instructions. So it takes longer for a digital organism to replicate. So there is where you have that trade-off. a digital organism might be better competing with others if it's not infected by a parasite, for example, but in order to, for the parasite, if the parasite wants to track changes in the bacteria and wants to evolve a new trait, wants to compute a new Boolean operation, that has a cost in terms of replication rate. It takes longer for it to make a copy. So, and that trade-off is something that emerged naturally here. And you also, in general, requires longer genomes for a digital organism to be able to compute some of these Boolean operations. Some of them are quite complex, like the equal function. And for performing those functions, the organism must execute a lot of instructions, many times the same instructions, or increase genome size so that it can jump from one instruction to the other more easily. So we are exploring those trade-offs.
Hi, sorry, just was wondering if in these kind of in silico models, does demography also play a role? Because like in natural populations, the decreasing number of individuals or can lead to different dynamics, but maybe with I don't know if the silica model takes that into account, or you just have a fixed number of individuals in each generation.
You have a carrying capacity and you find the same ecological dynamics that you could find in a purely ecological scenario. For example, and that's something I did, if you turn off mutations that you could do, because you just set your probability of mutation 0, you observe for this particular system the Lotka-Volterra cycles. So this system combines ecological dynamics and coevolutionary dynamics. And you can, and that's related with the discussion group that we had the other day, you can turn off, for example, the evolutionary component and work only on a purely ecological scenario. You can turn off ecology and work only on an evolutionary, purely evolutionary scenario. And you can explore the reciprocity between the two processes. But yes, population abundance plays a key role here.
Oh, thank you.
So you can also modify the carrying capacity by allowing more or less resources to be available for hosts.
Thank you.
In the same way our collaborators did in that previous work that I was showing you, where we explore how coevolutionary dynamics shapes network structure. They did a treatment with a lot of nutrients and the same experiment in a medium with only few resources. No? We can do the same. In fact, we are replicating that study in the lab in silico.
Okay, that was a second question. That
Yes.
if you were able to replicate that and
Yes.
get the same result. Thanks. Thank you very much. Do we have any other question? Yep.
Okay. So out of curiosity, how do you keep a virus static or a parasite or pathogen? So if you find that is able to counteract the resistance
Yes.
of future bacteria or whatever in the real application, imagine that this is going forward and in the end it has some applications. How do you keep, maybe viruses is easy, you keep them and I don't know, frozen or whatever, but what I mean is if you find a way to harness evolution, if
Yes.
you want to keep individuals that are more, they have a higher evolvability, okay, you want to keep that.
Yes, that's the idea.
How would you do it? I mean, it's
You frozen
in
them,
silico,
yes.
yeah, but I mean, we are thinking about this specific example, but I'm thinking about all the, I don't know. And it's also related to the question of how do you measure evolvability in the real
Yes,
world?
yes,
Because it's easy to measure that in silico, but
yeah.
you were talking about those viruses, they have a higher ability to infect future populations of bacteria. Doesn't mean that they are more evolvable, or they have a higher evolvability, because that's depending on the additive genetic variants they have. So maybe they have that trait, they are not more evolvable in that sense. You know what I mean?
Yeah, but let's say that you perform many coevolutionary experiments, and after 1,000 generations, you take samples of the phages, and then you can test the efficacy of that phage against a bunch of bacteria that you have previously isolated from other experiments, and see the fraction of bacteria that have evolved independently in different experiments are still susceptible to that phage. Could be a measure of evolvability, for example. If the phage that you take from your experiment is only able to infect the bacteria it has been co-evolving with, but it's not able to infect a bacteria that has evolved in an independent experiment, the evolvability of that phage would be lower than one that could do it.
It's a measure of past evolvability in a way. The question will be, is past evolvability predictive of a future evolvability? Which
I see. I see. Yes.
I guess it is, but I don't know. Those are some kind of maybe interesting questions. Well, and I just want to subscribe what Xavi said. I think this is very impressive work, and yeah, it's very welcome, and I think it's fantastic. So
there is now, this paper came out 1 month ago, I think, by Justin Mayer, and it's the first proof that how you can pre-adapt a phage so that it can infect the bacteria that you will have in a near future in the lab. These are real bacteria and phage. It's E. coli and the lambda phage. And there's a lot to be done on this, but I think this paper proves that this topic is worthy. We have to explore this because you can do that. They did it here.
Okay, maybe that's the.
So the point is to train phages, like in the movie, no? How you train your dragon here, how you train your phage, so that make it better able to cope with the resistance of the bacteria. Yeah. And I think we should try to put some effort on this topic because it also strategically, it also allows us to apply for calls on the biomedical area as well. And this is ecology and coevolution. It's predator-prey at the end. And it's ecology, it's competition. You can play with the competition matrix. And so we as ecologists, I think we have a lot to say on this topic.
I agree. Thank you.
Thank you very much. We have another question from Christoph. Which next? Okay, thank you everyone. We are going to the end of this session. You can still translate your questions to pass your questions to Miguel through his email. We don't have any online question. So thank you very much to everybody, and see you next week in the next EBD seminar session. Thank you very much.